A-Level Biology 9700, Paper 5, Variant 1, May, June 2024, and question number 1. Question number 1 is, a student investigated the effect of wind speed on the rate of transpiration. The student used the flowering plant Japanese Spirea japonica. The student used the apparatus shown in figure 1.1 to measure the rate of transpiration. And student, you can say the see the apparatus setup. The apparatus or the plant was set up into a container and having soil in it, and airtight seal is there and plastic bag around the container, and all the status was kept on the balance to find out the initial mass or final mass, and stop clock for a time period, and fan was used to give the wind speed to the plant. So to set up the apparatus, the student contained a Japanese pioneer plant growing in a container of soil, added 200 centimeter of water into the soil, placed a plastic bag around the container of soil to prevent water loss from the soil, placed the plant and container on the balance, switched on the fan to a low setting. The roots of the Japanese spirea plant absorbed water from the soil. Water was carried in the xylem and water vapors were lost by transpiration from the leaves of the plant. This caused the reading on the balance to decrease during the investigation. So identify the independent variable in this case. And the independent variable was wind speed, and due to which the rate of transpiration was changed, as the rate of transpiration was the dependent value. So the independent value is the wind speed. So the next question is, the student carried out the investigation in the laboratory with standard laboratory apparatus. Standard laboratory apparatus, the same apparatus was used for this purpose as it was used in the start of this experiment. Describe a method using the apparatus shown in figure 1.1 that the student would use to investigate the effect of wind speed on the rate of transpiration by Japanese pioneers. Your method should be set out in a logical order and be detailed enough to allow another person to follow it. So details how to set up the apparatus shown in figure 1 should not be included. So we are not going to include how we will set up the apparatus, but we will only tell the details about that this apparatus which was set for this purpose, we will give a detailed account on it. That is, first of all, I would like to tell you the details of this experiment, then we will write on the given space. So for this purpose, you can say we can use well-watered plant, as it has been shown into the figure. So we will set up the separators into an enclosed chamber with a constant temperature and light intensity, because in this investigation, the light intensity and temperature should remain constant. Otherwise, there will be a change in the rate of transpiration due to high or low temperature, due to high or low light intensity. So it should not be changed. So to keep them constant, we will use an enclosed chamber, and then we will use a fan uh, to give different wind speeds. So different wind speeds can be started from, we can give at least 5 different wind speeds. So we will give the 5 different wind speeds. We will start from the lowest speed, and then after every 1 hour, we will change the speed, and then we will find out the initial and final volume for each wind speed. So the initial and final volume will give us the values that how the rate of transpiration has been taken place. And sometimes you can say we can also use an anemometer to find out the or to measure the wind speed. But I think in these type of laboratory experiments, we can have only to show the initial and final volume or the mass in the given apparatus. So now we will start writing, and then we can repeat this experiment to find out the accuracy, or by taking the mean of all these repetitions. Now I would like to write this well-watered plant can be used for this purpose. So we will write down in the form of well-watered plant can be kept in an enclosed chamber where the light intensity and temperature can be kept constant. And student one more method can be used for this this purpose. When we keep this apparatus into a room, we can then we can have a light intensity. We can use a lamp to give the same light intensity or to provide the same temperature. So light intensity should not be changed because we will not use the natural light in this case, because if we are providing the lamp, so in every investigation and every measurement, we can have the same temperature and we can have the same light intensity. So we will keep on this experiment in this way, or then in this way we can maintain the temperature and we can maintain the light intensity. Then we will start with the lowest wind speed. Wind speed by using a fan and initial and final mass will be measured for this investigation. For about 1 hour in each case. So in this way, we will also keep the time constant. Then we will provide 5 different wind speeds by using the same fan, but every time for different wind speeds. Every time wind speed will be increased for this purpose. So at the end of this, after 1 hour, we can also repeat the investigation to find more accurate result. By calculating the mean. And there should be some um safety precautions. And every case we will make use of gloves, and we will wear the lab coat. And so in this way we can have the different uh, you know uh, problems to our skin, because we are using the soil as well. And in this way we can have we can have some hazard in this experiment. So we have to uh, keep everything in a way that the it's accurate results we can get in this way. So it was all about this investigation. Now we are moving towards the next question. And next question is, the student used a different method to measure the rate of transpiration of Japanese pioneer. Figure 1.2 shows the apparatus used. The leaf remained attached to the plant during the investigation. Lower side view and the side view, you can see the elastic band, two glass microscope slides, and lower surface of leaf. And they have used the cobalt fluoride paper, and the leaf is on glass microscope slide, plastic band, and glass microscope slide. When cobalt chloride paper is touched with the moisture, so its color is changed to pink. So piece of blue cobalt chloride paper was attached to the lower surface of the leaf as shown in the figure 1.2. Blue cobalt chloride paper changes color to pink if water is added. The student measured the time taken for the blue cobalt chloride paper to change color. This procedure was repeated with two more leaves of the Japanese spira plant. The plant was kept in controlled condition at all times. Show the results obtained using the cobalt chloride paper. So they have one, two, three leaf tested. There were three leaves tested for this purpose. And in the leaf one, 122, or the and the it was the time uh, which was taken for blue cobalt chloride paper to change color. It was taken into seconds. So in the second leaf, it was 137. In the third, it was 74. So by using the formula the student used to calculate the rate of transpiration in units, and the rate of transpiration is equal to 3600 over time taken in seconds. So we will, using the information given in the 1.1 and 1.3, calculate the rate of transpiration for leaf 2 of the Japanese spirilla plant. And for leaf 2, it is 137. So rate of transpiration will be, we will calculate the rate of transpiration in this way. Rate of transpiration 3600, and the value is 137 in the leaf 2. So it is about 26.3. By multiplying it with 10, we can get to 63. The unit will remain the same, but we are going to simplify it by multiplying it with 10. So it is very simple calculation which we have done for this question. And the next is, to improve the validity of the results, the student decided to measure the time taken for blue cobalt chloride paper to change color on a greater number of leaves of the Japanese spirea plant. State one other change in the student could make to the method to improve the validity of the results. So here you can see uh, we can have the we can make use of same sized cobalt chloride paper. we can use the same sized cobalt. So this is the one option. Okay. And we can use the, I think it is enough, 1 point is enough for this purpose. So it was all about the question number 1. Please like, share and subscribe my channel. Thanks for watching.